Hi, my name is Eliza Famador.
I am Bianca Jimenez.
I'm Mabel Babuji.
My name is Daniela Barrera.
And our model presentation is on animal cell and animal mitosis. This is the picture overview of the animal cell with all the organelles labeled onto the correct organelle. So I'm going to be talking about the nucleus and the mitochondria. There are three parts to the nucleus: the nuclear envelope, the nucleolus, and the chromatin. The nucleus is the control center of the cell. It spreads the genetic material and provides the instructions for protein synthesis. It is the largest organelle in the cell, and it contains fluid nucleoplasm, the nucleolus, chromatin, and is surrounded by the nuclear envelope. The nuclear envelope is a division between the cytoplasm and the nucleoplasm. It regulates passage of substances from in and out of the nucleus, and it is a double membrane structure with pores all around it. The nucleolus is the site of the ribosome subunit manufacture. The structure is a spherical, non-membrane bounded body that has ribosomal RNA and protein. The chromatin consists of DNA for the genes, a thread-like material that has histone protein DNA. Its primary function is packaging long DNA molecules into more compact, denser structures.
Next up, we have the rough endoplasmic reticulum. Appears studded with ribosomes, giving the rough ER its appearance and name. It sits next to the nucleus in the cell and is continuous with the nuclear envelope. Due to the studded ribosomes, the rough ER is responsible for the synthesis, modification, and transportation of proteins throughout the cell. The smooth endoplasmic reticulum, also known as the smooth ER, is a network of tubular membranous structures in eukaryotic cells. Although it is continuous with the rough ER, it is not studded with ribosomes, hence its smooth name and appearance. The smooth ER plays a major role in detoxifying the body from drugs, alcohol, and general metabolic waste, and serves as a site of steroid and lipid synthesis. Next, we have the ribosomes. Ribosomes are made up of two subunits, the large and small subunit. Both subunit contains protein and RNA. The location of ribosomes in the cell determines where the protein they make ends up. For example, free-floating ribosomes synthesize proteins to be used within the cell. On the other hand, ribosomes bound to the rough ER synthesize proteins to be used outside of the cell. The ribosomal function in the cell is to read RNA and synthesize protein. The Golgi apparatus is a stack of flattened membranes and a complex of vesicles within the cytoplasm of eukaryotic cells. It is located next to the endoplasmic reticulum and near the cell nucleus. The Golgi apparatus is responsible for transporting, modifying, and packaging proteins and lipids into vesicles for delivery to their targeted destination. Due to this function, it is most commonly referred to as the post office of the cell.
In animal cells, peroxisomes protect the cell from its own production of toxic hydrogen peroxide. As an example, white blood cells produce hydrogen peroxide to kill bacteria. The oxidative enzymes, catalase and oxidase, break down the hydrogen peroxide into water and oxygen. Next is a lysosome, a membrane-bound cell organelle that contains digestive enzymes. Lysosomes are involved with various cell processes. They break down excess or worn-out cell parts and may be used to destroy invading viruses and bacteria. If the cell is damaged beyond repair, lysosomes can help it to self-destruct in a process called programmed cell death or apoptosis. The centriole is a cylindrical structure composed of microtubules. In the cell, centrioles aid in cell division by facilitating the separation of chromosomes. For this reason, they are located near the nucleus. Centrioles are also involved in the formation of cilia and flagella, and thus contribute to cell movement. The flagellum is found on the outside of the cell and functions for movement. The flagellum are long, tail-like structures on the outside of the cell and move by whipping back and forth. The cytosol serves several functions within the cell. It is involved in a signal transduction between the cell membrane and nucleus and other organelles. It transports metabolites from their production site to other parts of the cell. It is important for cytokinesis when the cell divides in mitosis.
Moving on to the cytoskeleton elements, this part of the animal cell structure consists of the plasma membrane, microtubules, microfilaments, and intermediate filaments. These elements are vital to the animal cell because they not only provide structure and support for the cell, but they aid in essential cell functions. Starting with the plasma membrane, its main function is to act as an external barrier and is composed of phospholipids, globular proteins, and sugar chains. However, it also facilitates what comes into and out of the cell and is selectively permeable. External facing proteins also act as receptors for hormones, such as hormones, neurons, and muscle cells. The membrane also plays a vital role in signaling and cell-to-cell interactions. Microtubules are composed of tubular proteins and are cylindrical shaped. The main function is for cell support and to provide cell shape. It is also involved in cellular movement. Microfilaments are made of actin proteins and are fine filaments twisted around each other. It helps form the cell's cytoskeleton and is involved in muscle contraction and some cell movements. Intermediate filaments are mainly composed of protein fibers, and the shape depends on the different function of each cell. These filaments resist mechanical forces that try to come upon the cell. They are also known as the stable elements of the cytoskeleton.
This is our model presentation on the animal mitosis cycle. As you can see, a cell spends the majority of its time in interphase, followed by the mitotic phase, which includes prophase, metaphase, anaphase, and telophase. Afterwards, the cell goes into cytokinesis, which is the separation of the cytoplasm, resulting in two identical daughter cells. However, we would like to clarify that interphase and cytokinesis are not part of mitosis.
To start off the mitotic cycle, during prophase, the chromosomes first begin to appear. Next, the nucleus membrane begins to break down, and the centrioles produce mitotic spindle fibers.
Next, the metaphase stage of mitosis is a short process and doesn't last very long in the mitosis cycle. Chromosomes line up on the equator, also known as the metaphase plate, and prepare to be pulled apart in the anaphase stage.
During anaphase, the centrosomes push out and the microtubules are then shortened. The sister chromatids are separated when they were held together by the centromere.
The final stage is telophase. A few important things begin to occur during telophase. First, as the spindle fibers disappear, the chromosomes that appear during prophase now begin to uncoil and resume chromatin form. Secondly, the nuclear membrane that dissolved during prophase now begins to reform. And lastly, a nucleoli appears in each of the daughter nuclei.
Thank you.
This concludes our presentation. Okay, bye.